Welcome to AP Biology. This video is on proteins and nucleic acids. The monomer of a protein is called an amino acid. All amino acids have a very similar structure. All amino acids have a central carbon. Bonded to that central carbon is an amino group, NH3, that you see right here. Bonded to the central carbon is a carboxyl group that you see right here. And you have a hydrogen. These three groups are going to be same in every single amino acid. The group that changes from amino acid to amino acid is this group on the bottom called the R group. I like to think of it as the random group. There are 20 different amino acids total, and what makes one amino acid different from another is going to be this R group. It can make the amino acid polar. It can make the amino acid nonpolar. It can make it acidic. It can make it basic. It can make it neutral. The R group is what gives an amino acid all of its properties. If you look at all these polar amino acids, you can see that they're going to contain an oxygen. They can contain sulfur. They can contain nitrogen. These are all atoms that are electronegative and cause a distribution of electrons, making it a polar amino acid. If you look at all these nonpolar amino acids, you can see they're made up primarily of carbon and hydrogen, where all the electrons are equally shared. When two amino acids are bonded together, this is going to be done as usual through dehydration synthesis, the removal of water. You can see a bond form where the hydrogen of one amino group is going to react with the hydroxyl of a carboxyl group. You have the removal of water, dehydration synthesis, which forms a chemical bond. This bond is called a peptide bond, the bond between amino acids. Proteins are arguably the most important macromolecule within the body, and they are far more than just a simple polymer. They're not just a long sequence of amino acids peptide bonded together. They have a very complex structure. There are multiple levels to the structure of a protein. The most basic level of protein structure is called the primary structure. The primary structure is the linear sequence of amino acids peptide bonded together. The primary structure, or the linear sequence of amino acids, is ultimately what's going to determine the structure or the shape of a protein. Remember, the shape of the protein is what's going to determine its properties or its functions within the body. Now, if you look at this primary structure and you look at the individual amino acids, you can see that the oxygen of one amino acid is going to be attracted to the hydrogen of another amino acid. This is going to cause hydrogen bonds to form. And these hydrogen bonds that form are going to cause the linear sequence of amino acids to bend and twist and form a secondary structure. This secondary structure is made up of either alpha helixes or it's made up of beta pleated sheets. What's going to determine whether an alpha helix or a beta pleated sheet forms? It's going to be the linear sequence of amino acids. The sequence of amino acids is going to determine where the hydrogen bonds form and determine which one of these two structures is going to be created. Now, this sequence of amino acids is going to even further bond. We're going to see in the tertiary structure, the third level of structure, more hydrogen bonds form. You're going to see hydrophobic interactions. You're going to see disulfide bridges. You're going to see ionic bonds form, and it's going to form this complicated tertiary structure. Now, this tertiary structure that you see right here is going to be what we consider to be a polypeptide subunit. It's not a functional protein. Most functional proteins are made up of multiple polypeptide subunits put together. When you put together a number of these subunits, you form the quaternary structure, which is then going to be your functional protein. Again, what's going to create this quaternary structure, what's going to lead to the shape of this quaternary structure, this protein? Ultimately, it's the linear sequence of amino acids, the primary structure. Some proteins are made up of only 2 polypeptide subunits. We call them dimers. Some are made up of 3. Here you see an example of collagen, which we're going to talk about in a minute, made up of 3 polypeptide subunits. Kind of looks like a rope all twisted together. Some proteins are made up of 4 polypeptide subunits. Here you see hemoglobin, which we'll also talk about in a minute. There are 2 major protein shapes. You have fibrous proteins, which are structural proteins within the body. A good example of this structural or fibrous protein is collagen. You can see it looks like a rope twisted together. That is the major structural protein in the body. It is the most abundant protein in the body. It gives all of your tissues its strength. Another structural or fibrous protein is going to be keratin. Keratin is found in your hair. It's found in your nails. Another structural or fibrous protein is silk. This is what's found in a spider's web. We also have elastin. Elastin is what gives your connective tissues its elastic properties. The other type of protein shape is going to be the globular protein. These look like blobs. They're kind of more round in structure. Here you see hemoglobin, which is responsible for carrying oxygen and makes a part of the red blood cell. You also have microtubules, which are used to transport materials within the cell. You have enzymes, which speed up chemical reactions in the body. You have antibodies, which play a major role in the immune system. These are all globular proteins. Here we see a good example of how important the primary structure of a protein is. Again, it all boils down to that linear sequence of amino acids. A great example is found with sickle cell anemia. Sickle cell anemia revolves around a mutation in the hemoglobin protein. Here you see the primary structure of the hemoglobin protein, its linear sequence of amino acids. Ultimately, this is going to create a normal hemoglobin, which is going to make a nice biconcave red blood cell. A red blood cell that has lots of surface area that can be used to transport oxygen throughout the body. With sickle cell anemia, there is literally only a change in one amino acid in the primary structure. You can see that glutamic acid is changed into valine. We'll see later on this is due to a mutation in the DNA. By having a change in just one amino acid is going to lead to a change from this normally shaped red blood cell to this sickle-shaped red blood cell, which is not very efficient at carrying oxygen. You can see how just a change in one amino acid can lead to a change in the entire shape of the protein. Again, everything boils down to that primary structure, the linear sequence of amino acids. Proteins are going to perform all the major functions within the body. Proteins function as enzymes. Enzymes are used to speed up all of our chemical reactions. Proteins are used in the immune system, right? They are antibodies, which are going to destroy foreign invaders like bacteria and viruses. Proteins can be used to store amino acids. Proteins are used in transport. They are used as tunnels that allow large materials to enter or leave our cells. Proteins function as hormones within the body, used to communicate messages for one part of your body to communicate with another part of your body. Proteins are going to function as receptors. They're going to allow chemical messengers to transmit information into a cell. Proteins function as contractile and motor proteins, allowing cells to move or allowing movement within the cell. And proteins can also be structural, right? Giving hair, feathers, skin, all of its shape, its structure. A couple of factors can alter the structure of a protein. pH, changes in pH, either pH becoming too high or too low, changes in salt concentration or salinity, and changes in temperature. If the pH that a protein is in changes too dramatically, if the temperature is increased too much, for instance, if a protein is placed in boiling water, that protein will denature. To denature means that all the chemical bonds are broken and that protein is going to unravel. Once that protein unravels or denatures, it is then biologically inactive. It cannot do anything. Finally, we have our nucleic acids. This is just going to be a very simple, basic overview because we're going to cover nucleic acids much more in depth in our genetics unit. The monomer or the building block of a nucleic acid is a nucleotide. Here you see a picture of one nucleotide. Every nucleotide is made up of three subunits, three things. One is going to be a phosphate group. Two is going to be a sugar. That pentose sugar could be deoxyribose in DNA, or it would be ribose in RNA. DNA and RNA are the two types of nucleic acids. So you have a phosphate, the sugar, and finally you have a nitrogenous base. There are 5 different nitrogenous bases. You can have adenine, guanine, cytosine, thymine, and uracil. These nucleotides are bonded together to make the nucleic acid. Again, 2 types of nucleic acid being DNA and RNA. These 2 types of nucleic acids are going to perform different functions within the cell. The DNA is what carries the genetic code. It's the instruction manual on how to create all the body's proteins. RNA is going to be used to transfer the information from DNA to the ribosomes, which are going to be the site of protein synthesis itself. This concludes this video. Thank you.